Good morning, my dear students. Welcome to our Zoology online class. Dear students, in the last class, we studied about two important imaging techniques. Okay, two important imaging technique. One is the PET and the second one is MRI. Okay, PET full meaning the positron emission tomography scanning. Okay, positron emission tomography scanning, and the second one MRI, magnetic resonance imaging. Okay, so these two imaging techniques already we have completed in the last class. Okay, the working mechanism and the clinical significance of these two instruments. Then in the last class we also completed one more instrument, therapeutic instruments. Okay, therapeutic instruments, example we studied about the pacemakers. So the working mechanism and the uses and the structure of these pacemakers also we have completed in the last class. Understand, my dear students? So today we are going to study about one more therapeutic instrument. Okay, today we are going to study about one more therapeutic instruments. The instrument and name is called medical laser. The instrument name is called medical laser. Understand? So this device, medical laser device, is producing or releasing electromagnetic radiations. Okay, so this laser, medical laser, releasing this device, releasing electromagnetic radiations, and these radiations are used in many purpose in the clinical field. Okay, used in the clinical field for many purpose. For example, to cut the tissues, to coagulate the blood, to remove the tissues, damaged tissues. Okay, like this, some for many purpose these lasers. Okay, these medical lasers are used. Understand, my dear students? Understand? So medical laser is a device, a release or emit electromagnetic radiations. These radiations are used to cut the tissues, damaged tissues, are used to coagulate the blood, or used to remove the damaged tissues. Understand? Here, laser. Laser stand for light amplification by stimulate emission of radiation. LASER, laser, the full form laser is light amplification by stimulated emission of radiation. Okay, this is the full form of laser. Understand? Understand a few? Then maximum in surgery. Okay, this lasers is used. The laser used in the surgery field is non-ionizing radiations. Non-ionizing radiations. Understand, my dear students? Actually, the light emitted or released from a bulb. Okay, the ordinary light emitted from a bulb has many wavelengths, and this light can spread in many directions, in all directions. Okay, the ordinary light released from a bulb has many wavelengths and it can spread in all directions. Understand? But the laser light has only a specific wavelength. Okay, only a specific wavelength, and this light can spread in a narrow direction. Okay, in a particular single direction only, and create very high intensity. Okay, so the radiations it has only a specific wavelength, so it can focus only a particular direction. Understand a few? Then this medical laser is also controlled or guided by some computer based devices. Okay, so by using computer based devices, these medical lasers are used in the clinical field. Okay, used in the clinical field. Understand a few? Then maximum these lasers are used for cutaneous and ocular applications. Here cutaneous meaning skin related problems, ocular meaning eye related problems. Okay, so the maximum the medical lasers are used in the cutaneous and ocular problems. Understand, all of you? Understand? Then this laser also used in different types of endoscopies. Okay, endoscopy is an imaging technique. Okay, then bronchoscopy, laparoscopy, like this many different types of endoscopy techniques also this laser light are used, or medical lasers are used. Understand, my dear students? Understand? So now we completed one therapeutic instrument. Name of the therapeutic instrument is called medical laser. Okay, medical laser. So this medical laser is used to cut or remove or coagulate some damaged tissues from our body. Here the laser stand for light amplification by stimulated emission of radiations. Understand if you understand my students? Okay. Then next we are moving to next technique, biomedical technique. Okay, now we are going to study about one important techniques, blood cell counting technique. Okay, blood cell counting technique. So here in this technique one device is used. Name of the device is called hemocytometer. So hemocytometer is a device used to count the blood cells. Blood cells means you all know RBC, WBC, platelet. These three are the important blood cells present in the blood. So these blood cells can be counted with the help of this hemocytometer instrument. Understand my dear students? Now first I am going to explain about the structure of this hemocytometer. Okay, structure of hemocytometer. Listen very carefully my dear students. See, hemocytometer is a thick glass slide. Okay, thick glass slide. It consists of a chamber in the middle. Okay, it consists of a chamber in the middle. Name of the chamber is called counting chamber because this chamber only used to count the blood cells, whether RBC or WBC or platelet. Okay, so hemocytometer is a thick glass slide. In the center region, a counting chamber is present. Understand? And different types of counting chambers are available in the medical field, but maximum the commonly used counting chamber is improved Newborn counting chamber. Improved Newborn counting chamber is a counting chamber mostly used in the clinical field in hemocytometer. Understand all of you? Understand? Then listen very carefully. This counting chamber has 3 squares, 3 by 3 squares. Okay, 3 millimeters sized squares are there. These squares are called primary squares. Okay, so this improved Newborn counting chamber has 3 by 3 millimeter primary squares. Then next point, listen, the primary squares are further subdivided into 9 secondary squares. So 3 by 3 millimeter sized primary squares are there. So 3 primary squares are there. So these primary squares are again divided into 9 secondary squares. And size of these secondary square is 1 millimeter. Okay, 1 into 1 millimeter. Understand all of you? Understand? So the primary square size is 3 into 3 millimeter. Then the secondary square size is 1 into 1 millimeter. Understand all of you? Then the 4 corner squares are used for the white blood cells count. Okay, so the squares present in the 4 corners of the counting chamber is used for white blood cell count. Okay, so the white blood cells are count by using the 4 corner squares present in the counting chamber. Okay, so these chambers, the chambers which are used to count the blood cells are again subdivided into 16 tertiary squares. 16 tertiary squares. Understand my dear students? So primary square, 3 by 3 millimeter size primary square. Then the 3 primary squares are again divided into 9 secondary squares. The size of the secondary square is 1 by 1 millimeter. Then the 4 corner squares are used for counting the WBCs. These 4 corner squares are again subdivided into 16 tertiary squares. Okay, then the central secondary square. So the 4 corner squares only used for the counting, used to count the WBCs. Okay, so the secondary square present in the central region is divided into 25 tertiary squares. So the secondary squares present in the 4 corners are subdivided into 16 tertiary squares and they are used to count the WBC. Okay, then the secondary squares present in the central regions are subdivided into 25 tertiary squares. Okay, so the size of this 25 tertiary square is 0.2 by 0.2 millimeter. 0.2 by 0.2 millimeter. Understand, dear students? Then each single tertiary squares is further divided into 16 smaller squares. 16 smaller squares. Understand, all of you? Understand? Then the 5 black color squares and the shaded squares present in the center region are used to count the platelets. Okay, so 5 black color squares and the shaded squares which are present in the center region is used for the platelet count. Understand, all of you? Then 5 black color squares are used for a red blood cell counting. Red blood cell RBC counting. Understand, my dear students? So the corner squares. Okay, the corner squares are used for the WBC counting. Then the black color squares and the shaded squares which are present in the center region of the counting chamber is used for platelet count. Then 5 black color squares are used for the RBC counting. Understand, my dear students? Understand? So I hope you all understood the structure of this counting chamber. So once more I repeat all the points about this counting chamber. So counting chamber, improved Newbar counting chamber is the commonly used counting chamber. Okay, so this counting chamber is divided by 3 by 3 millimeter sized primary squares. Then the primary squares are further subdivided into 9 secondary squares. Size of the secondary squares is 1 by 1 millimeter. Then the 4 corner squares are used for the white blood cells counting, which are subdivided into 16 tertiary squares. And the secondary squares which are present in the central region is again divided into 25 tertiary squares. The size of the square is 0.2 by 0.2 millimeter. Okay, then each single tertiary squares are further divided into 16 smaller squares. Understand, all of you? Understand? Then listen, students, to count the blood cells, the blood should be, before counting the blood cell, the blood should be diluted by using proper diluting fluid. For example, RBC diluting fluid name is called Haim's solution. Okay, Haim's fluid or Haim's solution is used for RBC dilution. Then Track solution is used for WBC dilution. Okay, so before counting the blood cell, the blood should be diluted by using appropriate diluting fluid. Here Haim's solution is the RBC diluting fluid and the Track solution is the WBC diluting fluid. Understand, all of you? So when we apply this Haim's solution in the blood, this Haim's solution destroy all the other blood cells except RBC. So when we apply the Haim's solution, this Haim's solution lyses or destroy the WBC cells and platelets. So the RBCs are not lysed. Okay, understand, all of you? Then the blood is diluted 1:200 times with RBC diluting fluid. Then the cells are counted under the 40x objective of the microscope. 45x lens of the microscope. Understand, of you? Then second one, to count the WBCs, already I told you Track solution is used. So when this Track solution is added in the blood, this Track solution destroy or lyses the RBC and platelets. Okay, RBC and platelets. So we can get WBCs only. So by using this fluid, we can dilute the blood. So finally we can get only WBCs. Understand, of you? Because this Track solution consists of an acid called acetic acid. Okay, it consists of an acid called acetic acid. Understand? And WBC blood is diluted 1:20 times. Okay, and these WBCs are visualized by using 10x lens of the microscope. 10x lens of the microscope. Understand, my dear students? So there are two types of fluids are used for dilution. One is RBC diluting fluid and second one is WBC diluting fluid. RBC diluting fluid is called Haim solution and WBC diluting fluid is called Trex solutions. Understand all of you? Understand? Then separate pipettes are also used to count the blood cells. To count the RBC, separate pipettes are used, and to count the WBCs also, separate pipettes are used. Understand, my dear students? Understand? Then, say, next, to count the blood cells, first we have to collect the blood till 0.5 level or 0.5 graduation in the peptide. Okay, so to count the blood cells, the blood is taken till 0.5 in the pipette. Understand all of you? Then diluting fluid is taken till 11 to 101 level of the pipette. So to count the RBC, if you want to count the RBC, we have to take the RBC pipette first. So 0.5 level the blood is taken. Then 11 to 101 level the RBC diluting fluid. That means the Haim solution is taken. Understand all of you? Then the blood is diluted and mixed by rotating the pipette horizontally several times. Okay, so in the pipette blood is there and also the diluting fluid is there. Both should be mixed properly. For this purpose, the pipette should be rotated in horizontal direction for many times. Understand all of you? Understand? Then the tip of the pipette is placed on the counting chamber. And the fluid, here fluid means blood and the diluting fluids, okay, slowly released till it fill in the chamber. Okay, tip of the pipette is placed on the counting chamber, okay, placed on the counting chamber, and the fluid is slowly discharged or released till it fill the chamber. Understand all of you? Then cover slip is placed on the top of the counting chamber. Cover slip is placed on the top of the counting chamber, and we have to wait for a few minutes to settle all the blood cells in the counting chamber. Okay, then by using the microscope we can count the blood cells. By using the counting microscope we can count the blood cells. Understand, my dear students? So the steps involved in this are blood taken. So the first point, the blood is filled 0.5 levels of the pipette. Okay, then 11 to 101 level of the pipette, the diluting fluids are filled. Understand all of you? Then the blood and the dilution fluids both are mixed properly by rotating the pipettes in the horizontal direction for several times. Cover slip is placed on the top of the counting chamber. Then from the pipette, the blood is or the solution is slowly discharged inside the counting chamber. Okay, then we have to wait for a few minutes to settle all the blood cells. And by using the microscope, we can see we can count the blood cells. Understand, my dear students? Understand? Then the steps involved or the methods involved in the preparation of blood smear. Okay, steps or methods involved in the preparation of blood smear. Listen very carefully. To prepare a blood smear, we have to take 2 glass slides. So, in the first glass slide, we have to keep a drop of blood. Okay, we have to keep a drop of blood 1 centimeter from one end. So 1 drop of blood is kept in one slide from 1 centimeter distance from one end. Understand all of you? Then using the second glass slide. Okay, the second glass slide is one end of the second glass slide is placed on the blood drop about 45-degree angles. Okay, so in the first slide, 1 drop of blood is kept 1 centimeter distance from one end. Then one end of the second slide is placed on the blood drop about 45-degree angle. Okay, now the third step, spread the blood drop quickly on the outer surface of the first slide. Okay, spread the blood drop quickly, okay, quickly to make a small film, Then fourth step, a stain should be applied. Okay, Leishman stain. Okay, Leishman stain is the colored substance is added in the blood now. Understand? Then allow the slide to dry. That means we have to wait few minutes for dry. Understand? Then wash all the excessive stains, and finally keep the slides under the microscope, and we can easily we can easily understand the blood cells. Understand, my dear students? So once again repeat the steps or the methods involved in the preparation of blood smear. So we have to take 2 slides. In the first slide, from one end 1 centimeter distance, 1 drop of blood is kept. Then the second slide is, one end of the second slide is placed on this blood drop. Okay, placed on the blood drop about 45-degree angle. Then the third step, spread the blood drop quickly and make a small film. Then a color substance, Leishman's color substance, is added in the blood. Is all of you? The next step, allow the slides to dry. Few minutes we have to wait to dry the slides. Then wash the excessive stains, and under the microscope we can observe the blood cells. Understand, my students? Understand? So dear students, in this class we have completed one important therapeutic instrument, medical laser. Okay, the uses of the medical laser and the working mechanism of the medical laser we studied. Then one biomedical technique, okay, blood cell counting techniques also we have studied. Okay, here the instrument used to count the blood cell is hemocytometer. We have completed the structure of hemocytometer. We have studied about 2 diluting fluid and some pipettes are there. Then we have studied about how the blood is taken and how the diluting fluids are taken in the blood. That steps also we studied. And finally we studied about the blood smear preparation methods also we studied. Understand, my dear students? Understand? See, maximum there are 5 types. Okay, not maximum, our blood consists of 5 types of WBCs. Okay, neutrophil, eosinophil, basophil, monocyte and lymphocyte. Okay, so maximum 50 to 70% of the WBC is neutrophil. Okay, 50 to 70% of the WBC is neutrophil. 20 to 40% of the WBC is lymphocyte. 20 to 40% of the WBC is lymphocyte. Then 2 to 8% of the WBC is monocyte, 1 to 4% is eosinophil and 1% basophil. Okay, 5 types of WBCs are there, and the percentage of this WBCs also now be completed. Understand, my students? Okay, dear students, today's class also very useful to you. Hope you all enjoy today's class. Okay, dear students, thank you. We will meet in the next class.